previous video, we talked about alignments of bacterial sample reads against a reference genome, in this case, Pseudomonas aeruginosa. And what I want to do in this brief demo is show you how to save the variant calls to an Excel spreadsheet and interpret that a little bit. So I already have the alignment viewer set up in Geneious, and we're basically showing the reference genome and the aligned reads to it. And we're also set to the first locus in the genome, and so we're all set to go here. Looks like down here we have the list of all the variants that were called during this particular alignment. Over on the right we have the CDS or coding sequence checked, and we also have the variant track checked, so we can look at all the variants that are in this alignment. So what we can do, a couple things here, we can go down and click on this small right arrow, and that'll take us to the first polymorphism, or SNP in this case. And what it's showing here is that we have cytosine in the reference genome and guanine in the sample read. If we hover over these, we can get a metadata about the actual SNP itself. And then this is also coordinated with a table down below that shows the SNPs in more detail, the polymorphism in more detail. Basically, it's showing we have a SNP here, transversion, which is basically from cytosine to guanine. So this is going to be from a pyrimidine to a purine. And so that's the transversion. This is also intergenic because it's not showing the actual gene above here. If we go to the next variant, we see this one's actually occurring in a gene. It gives the gene name. In this case, it's some kind of a hemolysin gene. And it's showing that this is a transition type of SNP, in this case from pyrimidine to pyrimidine, thymine to cytosine. And we can just scroll through these if we wanted to, every single SNP that's out there, every polymorphism or variant that's out there. One thing we can do, however, is we can set up this table. We can customize this table any way we want. So for example, if we click on the columns header here, it shows all the columns that are available to you. If we do manage columns, it shows the available fields that are in there. And we have selected just a subset of those fields because in some cases these are more important than others. And I'll explain what these are when I get to this step where we're looking at the actual Excel spreadsheet. So I'll just show you that we just pull these from the available column over to the selected column, click OK, and then these fields show up here. Now one thing you can do, which is pretty nice, is you can export this table. Just click Export Table, and it'll save it as a CSV file. And so we'll just go ahead and do that. And that's saved it as a CSV file in one of our directories. And then what I'll show you next is how to import that into a Excel spreadsheet and look at it. Okay, so here is the spreadsheet that I was talking about. What we've done is we've imported the CSV file into Excel, reformatted it a little bit to make it look better, and then saved it out as an Excel spreadsheet. And then what we have here is the variant call list. So let me just go through the different fields that are here. The first one is the name field. Unfortunately, the default naming in Geneious is not very useful. So what we normally do is we will numerically identify these and then order them like we see here. The next two fields, min-max, that's the genetic loci, genomic loci. And we've ordered these along the minimum field. So it's increasing from the minimum. And then for each one of these records, it shows the length of the SNP. It can be just a single base change, or it can be multi-base change if it's going to be insertion, deletion, small indel, that sort of thing. The next field is the type of polymorphism that we see here. There's several different kinds. There's transversions and transitions. So transversion is going to be from purine to pyrimidine or vice versa. And then transition is from purine to purine or pyrimidine to pyrimidine. I think we have insertions, deletions, substitutions. I think stop codons show up here once in a while. And so all the different kinds of polymorphism types will be listed next. We then have amino acid changes. If there is an amino acid change, it'll show up in this list. Now the first one, there was no amino acid change apparently, so that's just left blank. However, if we go down to this one, we have the one-letter code for amino acids throughout this list. And so K to E, I think that's lysine to glutamic acid. you can get the one-letter codes online. So if there is an amino acid change, it'll show up here, and those can be pretty important usually. The next field is the coding sequence. And if it's blank, that usually means it's a intergenic type of polymorphism, so there's no name associated with it. Once in a while, the GenBank file itself that this is based on may not have an actual name in there occasionally, so that might be a problem if it shows up blank there. But if it's in a genic region, it'll show the name of the gene as it appears in the GenBank file. In this case, it's hemolysin or hypothetical protein, FHA domain containing protein, or whatever the gene name might be. Okay, so that'll give you the CDS name. Then, if there is a codon change, it will show up in this column, the codon change column. And so sometimes there is no codon change, and sometimes there is. And if there is a change, it'll show up here. And then the final column, and probably the most important, is called protein effect. If the actual codon change has some kind of structural or functional change in the protein, it'll show up here. So for example, there might be substitutions in an amino acid in a protein, there might be deletions, there might be frame shifts or something else that occurs in the protein or enzyme. Presumably, if those are deleterious changes, it might change the structure and then the function of the protein or enzyme that it's affecting, and that is usually a fairly serious problem. You'll probably want to spend most of your time looking at the protein effects, if there are any, and look at those polymorphisms first. So, and of course, you can filter and rank order these any way you want. And so that is basically how we generate the variant call list in Excel from our Geneious alignments.